Good afternoon, and welcome to the Immune Deficiency Foundation's Lunch and Learn series. Today we'll learn more about complement deficiencies, how they're managed, and resources for supporting families navigating this diagnosis. This presentation will be led by Dr. Eve Wu from the University of North Carolina, Chapel Hill. My name is Megan Messick, and I'm the Director of Education at the Immune Deficiency Foundation. On behalf of everyone at the Foundation, we want to thank you for tuning in to this afternoon's program. We recognize that through the provision of timely, comprehensive, and accessible information, that we can improve the diagnosis, treatment, and quality of life of people affected by primary immunodeficiency. And thank you for being a part of that journey today. Before we dive in, we do have a couple of housekeeping notes as well for you this afternoon. So the first is that we'll be using our Zoom webinar feature. Attendees should be able to see the slides and hear the presenter and the host speak. Attendees will not be able to activate their video camera or their microphone. There will be an opportunity today for questions after the presentation, and we kindly ask that you submit those throughout the session using the Q&A box in the control panel of your screen. Please ensure that those questions stay relevant to today's topic. All questions that we have time for this afternoon will remain anonymous and read out loud, and please ensure to not include any personal health information in those submissions. A brief disclaimer as well. Please ensure that the information that we see today, it is not intended to be substitute for medical advice, diagnosis, or treatment. We're here today as a trusted resource and a friend to provide you with information, but always ensure that you're seeking the advice of your physician or other qualified health provider when it comes to questions concerning a medical condition. Never disregard professional medical advice or delay seeking it based off of information presented at an educational event. I'm also very happy today to thank our sponsors. It's due to their partnership and contributions that we can provide programs like this for the PI community. Please join me in thanking the sponsors displayed on today's screen. And now I am so pleased to introduce today's presenter. So Dr. Eve Wu is an associate professor of pediatrics at the University of North Carolina, Chapel Hill, and specializes in allergy, immunology, and pediatric rheumatology. She's actively engaged in both clinical and translational research projects that involve a spectrum of diseases that affect the immune system, and she has a particular interest in complement deficiency disorders. Welcome, Dr. Wu, and thank you so much for joining us. I'll go ahead and stop sharing my screen so that you may bring up your slides.
Thank you so much. As I pull up my slide, I want to thank Megan for that kind introduction. I want to wish everyone a happy new year and good afternoon, and want to thank everybody for this opportunity to speak today. Okay, let me pull these up, go to the beginning. Let me get myself situated here. All right, I like to use the laser pointer. Okay. So first, I have to admit, when I was invited for this opportunity to speak about complement deficiencies, I thought, and I still think, that it's quite a daunting task. And the reason for that is something that I hope you will appreciate by the end of this presentation, which is that complement deficiencies are not just one single immune deficiency, but instead are a collection or group of immune deficiencies dependent on which complement protein is actually defective. And because of that, the approach that I took today was more kind of a 30,000-foot view from the sky, general overview of complement and complement deficiencies. I still hope that, even though it's a general overview, that there will be aspects about this presentation that hit home for you, whether you be a patient who's been affected by a complement deficiency or are a medical professional who are here to learn more. And so with that, these are my disclosures. The objectives for today: first, we're going to talk about what complement is, all of its important functions, including how it helps defend us against infections. You know, to me, you need to really understand how something normally works so that when it's missing, you can sort of understand why it presents the way it does. And then we're going to talk about studies that are available to us to evaluate patients that we suspect have a deficiency within complement, and then end with a review of complement deficiencies, results of those evaluations that we just went over, and some general thoughts around the management of complement deficiencies. But first, talking about complement and what it does for us to help defend against infections. The complement system is actually very important in our innate immune system. And our innate immune system is kind of the most rudimentary first line of defense that we have against infections. It can include physical barriers like our skin, which can help keep germs out of our body. But it can also include circulating proteins that kind of swim around in our body, like complement. Complement is also actually a very important helper in our humoral immunity. So humoral immunity is a little more specialized, and it's mediated by B cells and antibodies that these B cells make. And this is actually how the complement system gets its name. It's because it complements or helps boost our B cell or our humoral immune responses. The complement system consists of more than 30 proteins that are generally produced by our liver. There are some exceptions, but most complement proteins are produced by our liver. One of the things that I find fascinating is that the complement system is evolutionarily ancient. So there have been complement proteins that have been reportedly found in sea urchins like you see here, entities like called sea squirts. I've learned that these are over 300 million years old. So the fact that complement has been so conserved through time in different species, to me, really emphasizes just how important it is. But some might say I might be a little bit biased around complement, which is probably true. So when it comes to complement, something that I hope that you'll appreciate, normally complement proteins will circulate around in kind of this functionally inactive, intact precursor form that we call a zymogen. That's just our fancy word. And then complement can actually only be activated through a sequence of triggered enzymatic reactions. And these reactions are very much like a relay race with the passing of the baton. So it's not until a complement protein receives that baton that it can be activated. And part of that is on purpose, right? So we don't want complement running around active in our system, potentially causing damage. So instead, they circulate around inactive until they receive that baton to be called into action to help us with things like fighting off infections that have invaded our body. Now, there are actually 3 different pathways or arms by which complement can be activated. And so the first is the alternative pathway, which happens to be the oldest, and then the classical and lectin pathways. And we're going to talk a little bit about how each of these pathways can be activated. But some other things I want to point out first, as well as some general rules of thumb I want to go over when it comes to complement. The first thing I want to point out is you'll see here, no matter how each pathway gets activated or initiated, they all have in common this complement protein C3. And complement protein C3 actually happens to be the most abundant complement protein that we have in our system. And so the fact that it's important or essential or has a central role in all 3 pathways really underscores the importance of how it functions in our immune system and in our defense against infections, which we're going to go over. Some general rules of thumb, and this is really about the naming of the complement proteins. So complement proteins are typically noted by a capital C, followed by a number, which is supposed to kind of reflect the order in which they appear in the pathway. Of course, there are always exceptions. And so one exception is C4, which happens to occur right after C1 and then before C2. The other general rule of thumb is, and not necessarily represented on this figure, but that we will talk about later, there are complement proteins that are unique to the alternative complement pathway, and these are usually designated as factors. Again, we're going to introduce some of those a little bit later. And then the last rule of thumb is something that you'll appreciate is, like I mentioned before, complement proteins usually circulate around in these inactive, intact forms. When they're called into action, they're typically cleaved into sort of 2 smaller fragments. Each fragment is designated by either a lowercase a or a lowercase b, okay? So now we're going to first focus on how does the alternative complement pathway get activated. What's interesting about the alternative complement pathway is that it actually always has this kind of low level simmering degree of activation. And so that complement protein C3 that we mentioned previously actually sometimes just kind of spontaneously cleaves apart into a small C3A fragment that then goes out into circulation, and then a larger C3B fragment. And this C3b fragment, if there's a microbe like a bacteria or a virus that's nearby, can actually attach to its surface. Now, if C3b were to inadvertently attach to one of our own cells, what we would call a host cell, there are actually mechanisms in place to rapidly break it down so that it doesn't cause any activation on our own tissues. But when the C3b binds to or attaches to the surface of a germ, what it can then do is partner with or attach with factor B. This is where those factors come into play for the alternative complement pathway. When factor B and C3b pair up, there's a factor D that then can cleave factor B into those two smaller fragments, BA, which goes off into circulation, and then BB. And when BB and C3b partner together, they form what we call a C3 convertase for the alternative pathway. And we'll talk a little bit later about what that C3 convertase does. But first, to the classical pathway. So the classical pathway for complement gets activated when this C1q molecule actually recognizes antibody, could be IgG antibody, IgM antibody, that is bound to the surface of a microbe, again, like a bacteria or a virus. Now, the C1 is actually kind of reminds me of those old arcade games that have the arm that picks up a toy or a stuffed animal, but in this case, the toy is an antibody. But the C1 complex is actually a complex of proteins. So there's a C1q molecule that is partnered with or complexed with two C1r molecules and two C1s molecules. When this C1q portion of the C1 complex is recognized as antibody, it allows it to cleave complement protein C4 into two smaller fragments and complement protein C2 into smaller fragments as well. And the 2A fragment and the C4b fragment partner together, can attach to that microbial surface and form the C3 convertase for the classical pathway. Now, for the lectin pathway, maybe something you guys might appreciate is this little complex here, consisting of a mannose binding lectin protein, looks awfully similar to the C1 complex for the classical pathway, and that's true. So this mannose binding lectin, very similar to C1q for the classical pathway, is complexed with two each of these MASP proteins that you see here. And this mannose binding lectin piece with these kind of globular heads can actually recognize sugar residues like mannose that exist or kind of smatter the surface of different germs, bacteria, and viruses. And so similarly, when mannose binding lectin recognizes these sugar residues on germs, then it allows it to kind of cleave and activate complement protein C4 and C2, and then the smaller fragments C2A and C4b partner up together and form the C3 convertase for the lectin pathway, which, as you'll notice, is identical to the classical pathway C3 convertase. So what do these C3 convertases do? You can see here, regardless of how each of these pathways are activated, they all essentially converge at formation of their C3 convertases. The alternative pathways looks a little bit different than that of the classical pathway and the lectin pathway, but they all do the same thing, which is essentially they convert or cleave C3 into smaller fragments, C3A, which goes off into circulation and has some very important functions, and C3b. And C3b can do one of a couple of things. One is, just like it did up here at the start of the alternative complement pathway, it can attach to the surface of a germ. But one of its other important roles is that the C3b, regardless if it's produced by the alternative, classical, or lectin pathway, the C3b will actually attach back onto the C3 convertase to form the C5 convertases for the different pathways. So you'll see here, now there are 3 fragments that are attached together to make the C5 convertases for the pathways. And what does C5 convertase do? Well, it converts or activate complement protein C5, cleaves it into C5a, a smaller fragment that goes out with C3a out into circulation, and then C5b. And C5b is a very important fragment because what it does is it activates what we call the terminal complement pathway that is common to all 3 pathways, regardless of how they got activated. And what this terminal complement pathway does is C5b will partner with or attach to complement protein C6, C7, C8, and then a series of C9 molecules. And these form what we call the membrane attack complex. And it very much is like a pore or a peg that you can insert into the membrane of a cell. Could be one of our cells, could be the surface of a bacteria or a virus. And what happens is when this peg is inserted or this pore is inserted, it allows fluids to kind of rush in. So it's very much like having a siphon in like a water balloon. And if all this fluid is going in, eventually the water balloon is going to achieve this capacity and it's going to result in the balloon bursting. Or in the case of like a bacteria, it's going to result in something that we call cell lysis. And so the fluids come in, the bacteria reaches its capacity, and essentially it bursts or undergoes something called cell lysis. This formation of the membrane attack complex, regardless of how these pathways get activated, this formation of the membrane attack complex is one of the very important ways that complement helps defend us against infection. One of the other important ways complement helps defend us against infection is what we call opsonization. So we mentioned how C3b can be produced by the breakdown or cleavage of complement protein C3. Well, that C3b, we talked about how it can bind to the surface of microbes. Well, opsonization is actually derived from the Greek word meaning like kind of like a delicacy or a condiment. So when C3b tags these germs, it actually alerts our immune system and makes these bacteria or germs appear tastier for our immune cells to actually engulf or eat, or another word is called phagocytose, these bacteria, which you can see here. So this is another important way that our complement system helps defend against infections. And then another way, which we talked about, is when C3 and C5 get cleaved, they will produce these smaller fragments, C3a and C5a respectively. And C3a and C5a, our fancy terms for them is anaphylatoxins. And essentially what they are they're sort of like kind of a cologne or perfume to attract neutrophils and monocytes, so certain immune cells, to sites of inflammation or infection. They say, hey, you know what, come here, join the party, help us get rid of these bacteria that have invaded our body. So again, regardless of how they get activated, what these pathways have in common are kind of the 3 ways that they help defend against infection. Formation of the membrane attack complex, formation of the C3b molecule, which is very important at opsonizing or tagging germs to be eliminated from our body, and then the recruitment of other immune cells to sites of infection to help kind of get rid of the germs that have invaded. But complement also has some other very important roles. So as we mentioned before, complement gets its name because it helps to boost our humoral immune response. And it actually can do that through several ways. An example being, so B cells, which help mediate our humoral immune responses, actually have little receptors or antennas on their surface, so that if there is a microbe that's tagged with C3b, the B cell will actually recognize that and bind the complement or the C3b that is tagging the microbe. And by virtue of that, it can actually help boost the antibody response that B cell will have. And so that's just one of the examples of how complements our humoral immune responses. Another very important function of complement, which is very relevant when we start talking about complement deficiencies, is it helps get rid of waste. They're kind of like garbage collectors. So at any point in our bodies, excuse me, at any point in our bodies, there are cells that are being born, cells that are undergoing cell death. And when cells die, sometimes they leave behind this debris or garbage that needs to be picked up. The other way that complement helps to get rid of waste is sometimes when antibodies that are produced by B cells attach to proteins in our blood, they form what we call these complexes or immune complexes. And if those aren't cleared out, those immune complexes can actually deposit in tissues like our kidneys, triggering inflammation and injury and damage to our kidneys. So it's very important that these immune complexes get swept up and cleared out of our body. Now, similarly, if for some reason complement is missing, and they can't clear out those immune complexes, which then can deposit in the organs and cause inflammation. If complement's missing and doesn't sweep out some of our debris or garbage from our own host cells, that's really one of the underlying mechanisms for autoimmune diseases like lupus. And so autoimmune diseases are when our immune system reacts to our own body. And one of the triggering events is thought to be when we have our own self proteins that are kind of circulating around and don't get cleared out. There's sort of a threshold that's reached that triggers our immune system to kind of have a reaction to our own cell proteins. And that's one of the underlying mechanisms for autoimmune diseases, of which individuals with complement deficiencies are at higher risk, which we'll talk about a little bit later. Okay, I always say whatever goes up must come down, right? And that's true when it comes to complement activation. So complement regulation or regulation of complement is just as important. You can imagine if we let all that complement activation go on undeterred, then it can cause damage to our own host cells or organs. So complement regulation is very important to prevent any complement mediated damage to the host. You got to get back to status quo when it comes to complement. And complement regulation can actually occur through multiple checkpoints within a given pathway in order to suppress or quiet down that activation. But here are a couple or a few ways that complement gets regulated. So first, and probably one of the most clinically relevant regulators for complement is a protein called C1 esterase inhibitor or C1 inhibitor. And what it does for, let's say here, if you look at the classical pathway, C1 complex, like we mentioned before, it's a complex of C1q, 2 molecules of C1r and 2 molecules of C1s. Essentially what the C1 inhibitor will do is it jars apart that complex. And by jarring them apart, it kind of renders that C1q molecule not functional. And this C1 inhibitor actually does the same for the mannose binding lectin. We talked about how that MBL protein is complex with those 2 MASP proteins. And so it jars them apart, again, rendering that MBL protein non-functional as well. Another way that complement activation can be regulated or dampened is those C3 convertase enzymes that we talked about. They can actually be broken down. And so you see here the C3 convertase for the classical and lectin pathways, the C3 convertase for the alternative pathway. There are these complement proteins, very similar to C1 inhibitor, kind of jarring those pairings apart. And again, rendering the C3 convertase not functional for the pathway. A third mechanism is that there's a complement regulator called factor I. And what it can do is take complement fragments like C3b and C4b and just break them down into a bunch of little fragments that really don't do much of anything. And then another way is that formation of a membrane attack complex can actually be inhibited. So there's a protein called CD59 or protectin that essentially binds to C7, C8, and then prevents the pairing or attachment of complement protein C9 and therefore formation of that pore. So it kind of turns off formation of that membrane attack complex. And then one of the last ways that complement can be regulated is those anaphylatoxins that we talked about, the perfume, cologne, C3A, C5A. There are some enzymes that essentially can break them down and make them inactive. Okay, so now we've gone through a lot of material, how complement can get activated, how it gets regulated. Now we're going to talk about studies that are available to us to evaluate patients who may have complement deficiencies. And I like to kind of divide this up into two parts. So the first is just quantity. How much is there, right? So you can actually quantify different intact complement proteins. By far, the most used in clinical practice is going to be measuring complement protein C3 and C4 levels. Rheumatologists like myself tend to monitor these with certain autoimmune conditions. Lupus is going to be the prime example. Patients with lupus tend to have low C3 and C4 levels when their disease is very active. So these assays or tests are available in most hospitals, clinical laboratories, or medical centers. When it comes to quantifying or measuring other complement factors, those tests are available, but I usually think of those getting sent to more specialized laboratories, and that includes the Mayo Clinic laboratories and National Jewish Health. Both have the capability of measuring complement factors, as well as sometimes some of those activation fragments that we talk about, for example, like C3A or C3B. Beyond that, there are some complement factors and function that are available through research labs. So one way to evaluate complement is looking at the quantity, how much of it is there. Another way you can look at complement is actually its function. And there's sort of two parts when it comes to the function. So one is there are functional tests for each individual complement protein and regulator that we talked about in the first portion of our presentation. But one of the cool things, and I'm not going to go over the technicalities of these tests, is you can actually look at the function of those activation pathways that we went over. So the AH50 is looking at the functional or functionality of the alternative complement pathway. The CH50 is a functional test for the classical pathway. And then MBLF is the functional test for the mannose binding lectin pathway. And I like to think of these sometimes as, in some ways like a screening test for complement deficiency. And the reason for that is when you're looking at the overall function of these complement pathways, it requires that each of these complement proteins that are unique to these pathways have to be present and functional, right? So all the way starting from C3 down to the terminal complement pathway and formation of C5b has to be present for AH50. For your classical pathway, you have to have everything present and functional all the way from C1 down to the terminal complement pathway, which is C5b through complement protein C9. And similarly, for the mannose binding lectin function test, you've got to have everything from MBL all the way down to C5b through complement protein C9. From stem to stern, all those complement proteins have to be present and functional in order to have a normal result for a functional test for any one of these pathways. And we'll take a little closer look at this. I think by doing these screening tests, it can kind of help us hone in addition to how a patient presents, it can kind of help us hone in on maybe what complement protein is missing or not functioning. So for example, that C1 complex is unique, only as important for activation of the complement pathway. So you can imagine if C1 were absent, really it should only affect the result of our CH50 test, but not affect the results of our MBLF or AH50 test, right? Similarly, let's say if you had a deficiency in factor B, by being a factor, we know it's unique to the alternative complement pathway. If it's deficient, it really should only affect the AH50 result, and the CH50 and MBLF test should be normal. And then last, if the mannose binding lectin is deficient, then it really should only affect the results of the MBL functional test, and the AH50 and the CH50 for the alternative and classical pathways should be normal, okay? And this is kind of summarized here. So if you have a very low or zero CH50, classical pathway function, but your alternative is okay, you're really going to be missing proteins that are unique to the classical pathway. And this is specifically C1. For C2 and C4, you would have not only a low CH50, but also a low MBL function. If your AH50 is 0 or very low, but your CH50 is okay, then again, you're going to be missing those proteins that are unique to the alternative pathway, those factors, right? Factor B, factor D, or factor P. And then if your CH50 and AH50 are both 0 and very low, then you know you're likely missing or something's not functional with one of those complement proteins that overlap between all of those pathways. So that's that complement protein C3 or those complement proteins that make up that membrane attack complex or that terminal complement pathway. And then last, if your CH50 and AH50 are okay and your MBL function assay test results are low, then you know that might point you to a mannose binding lectin deficiency. Okay, so now that we've gone over kind of what complement is, what it does, how we can measure it, now we're going to talk about complement deficiencies. So generally speaking, complement deficiencies represent about 5 to 10% of all primary immune deficiencies. They're typically going to be inherited in an autosomal recessive fashion. This means that typically you will have a father that's a carrier, represented here by this blue affected DNA, and then a mother who's a carrier, also represented by this blue affected DNA. And there's a 25% chance, if mom and dad are both carriers, to pass this on to an affected child. There's a 50% chance for them to pass it along so that their child is a carrier as well. And then a 25% of having an unaffected child where both pieces of DNA inherited from mom and dad are entirely normal. Now, the exceptions are C1 inhibitor, which happens to be autosomal dominant. So this means that the affected child can, inherit the affected DNA from either mom or dad and doesn't need 2 bad copies, just 1 in order to have symptoms. And then piperidin is actually on one of our sex chromosomes. It's the X chromosome. So boys often tend to be more affected than girls. Another general rule of thumb is most carriers of complement deficiency, so for in this example, mom and dad, are actually going to be clinically normal or without symptoms. There are always exceptions, but for the most part, complement deficiency carriers are going to be overall normal. Now, when we think about the way complement deficiencies present, they're kind of larger or broader categories that I wanted to talk about. Not surprisingly, we spent much of the talk talking about how important complement is at helping to defend against infections. And so when it's deficient, one of the main ways it presents is recurrent infections. And that includes recurrent sinus and pulmonary infections, particularly with encapsulated bacteria. The most common encapsulated bacteria that we see is Streptococcus pneumonia, which is a very important cause of ear infections, sinus infections, and pneumonias. And remember, the reason for that is that C3b is such an important tag of bacteria, including Streptococcus pneumonia. So when it's absent, or something related to that C3b is absent, it's going to make us more prone to infections with certain bacteria like pneumococcus. Children and adults who have complement deficiencies are actually also uniquely have an increased susceptibility to infections with a certain type of bacteria called Neisseria. And the reason for that is there's something about our defense against Neisseria that is uniquely dependent on formation of that membrane attack complex and activation of that terminal complement pathway. So something that you'll appreciate is a deficiency in any one of these membrane attack complex complement proteins increases your risk for infection, particularly with Neisserial bacteria, a particular strain called meningococcus. So recurrent infections, definitely one of the common ways that complement deficiencies can present. Another way is autoimmune disease, and we kind of touched upon this early when we were talking about how complement is very important as garbage collectors, helps really dispose of waste. And so when complement is deficient and doesn't either clear out cell debris from cells that have died, or cell proteins that are complexed or immune complexed with antibodies, if those don't get cleared out, again, remember, there's that sort of threshold where then our immune system ends up having a reaction to self-proteins triggering an autoimmune disease. Lupus is by far the prototypical one, the most common autoimmune disease that our complement deficiency patients will be affected by. The other is kidney disease. As we mentioned before, sometimes when you have those immune complexes that then kind of deposit in organs like the kidneys, there are very specific kidney diseases that complement deficiencies can manifest with, which we're going to go over. So now that we've kind of talked about the broad categories by which complement deficiencies can present, we're going to go over sort of which belong to which categories. So the first category is the recurrent infections that can occur with or without the development of lupus. And the first is going to be your C1 complement protein. Now, again, we mentioned that C1 complex consists of C1q and C1r and C1s. C1q deficiency is quite uncommon. You're going to see here, we're looking at overall just total number of cases that are reported in the literature. And so very uncommon. But of those individuals who have C1q deficiency, more than 90% of them will develop an early onset lupus. And what I mean by that is most of the lupus I tend to see in rheumatology clinic are they're usually teenagers. And so lupus tends to affect women who are kind of post-pubertal or of childbearing age. And so if I see a younger child who develops lupus and maybe has some other features, well, then I might want to think about evaluating that child for a complement deficiency. In fact, C1q is the strongest genetic risk factor for the development of lupus. We talked about how that C1 complex can help bind to antibodies that are tagged onto microbe surfaces. So you can imagine if deficient, then that can result in an increased risk for infection, particularly infections with encapsulated bacteria like Streptococcus pneumonia. C1R and C1S deficiencies are very rare, more rare than the C1q deficiencies. When your C1 protein is totally absent, you can imagine that it's going to result in a near zero CH50 or classical pathway functional test, whereas the AH50 and the MBL function will be normal. Now, C4 deficiency is a complex one. It's actually quite rare to have a complete C4 deficiency. And the reason for that is, let's say if you think of each of these as kind of a string of DNA that you inherit, one from mom, one from dad, there are actually two complement genes on each of these strings of DNA that you inherit from mom and dad. So the likelihood of having all four genes knocked out is extremely rare. Now, having said that, for those patients that do have complete C4 deficiencies, again, 50–80% of them are going to develop either lupus or some other autoimmune condition like scleroderma, IgA vasculitis, particular types of kidney disease called membranous and IgA nephropathy. And then, not surprisingly, they can also develop infections with encapsulated bacteria. And then the last complement deficiency in this grouping is C2 deficiency. Now, compared to the others, C2 deficiency is actually common enough that we can estimate a frequency. And so we estimate C2 deficiency to occur about in 1 in 10,000 to 1 in 20,000 individuals. Having said that, most patients walking around with complete C2 deficiencies are actually asymptomatic. For those that do have symptoms, again, what's been most commonly seen or described are lupus patients. Maybe about half of them will develop lupus. And then again, those infections with the encapsulated bacteria, all of these will result in near zero CH50 functional tests. The next broad category that we're going to talk about are the Neisseria infections, particularly with that Neisseria bacterial strain meningococcus. The first of the complement deficiencies that can present with that are those terminal complement pathway proteins, or those that belong to that membrane attack complex, right? We talked about how formation of the membrane attack complex is particular in our defense against Neisseria infections. And so if you have a deficiency in any one of those, these patients are again at increased risk for Neisseria infections. Some can have very severe infections, but some can have kind of milder infections. And in between those infections, they can actually be otherwise quite healthy. You'll see here that C5 deficiency is quite rare. C6, C7, C8 also rare. We're talking about numbers of case reports, and they do tend to occur in more race and ethnicities compared to others. And then your complement C9 actually is not uncommon. And these patients can also develop Neisseria infection. So in addition to your CH50 being low, your AH50 is going to be low for all of these complement deficiencies as well. The other grouping of complement deficiencies that can present uniquely with an increased risk for Neisseria infections is actually your alternative complement proteins, those factors that we talked about earlier. So factor D, factor B, and properdin. You can see here factor D and factor B are quite rare. We're talking about only a handful of families that have been described. And what's interesting is that, for example, with the factor D family that had deficiency, not everybody had symptoms. So it suggests that maybe there's more than just the factor D deficiency that is putting them at increased risk for not only Neisseria infections, but also those encapsulated bacteria like pneumococcus. Because these are factors that are unique to the alternative pathway, only the AH50 will be near zero or low. And one thing I wanted to point out, the reason why with complement C9 deficiency, why the CH50 is low and not zero, is because the test can still be quite functional, even with C5 through C8 being intact. So that assay is still somewhat works, even if you have a complement C9 deficiency. Okay. The other grouping, these kind of sort of live in their own world as far as complement deficiencies go. The first is C3. So we talked about how important complement protein 3 is to all three pathways. So it's kind of the central protein for the complement system in general. Again, you're looking at it's overall very rare, just cases that have been described in literature. We talked about how C3 is so important at tagging those germs for elimination from our immune system. So these individuals tend to have very severe recurrent infections. Some will actually present shortly after birth. They are also uniquely susceptible to a type of kidney disease called membranoproliferative glomerulonephritis. This is a kidney biopsy of a patient who has this membranoproliferative glomerulonephritis. And patients who have C3 deficiency, because it's common to all three pathways, again, all functional pathway testing is going to be essentially near zero or absent. Another complement deficiency that kind of stands on its own is that mannose binding lectin. So mannose binding lectin is a little bit complicated. It's thought to be actually quite common. Many individuals who've been described to have mannose binding lectin. The challenge is there have been many individuals who've been found to have low levels and aren't necessarily symptomatic. It's also been hard to kind of establish what are normal MBL levels in otherwise healthy individuals. So MBL deficiency is a little bit nebulous for some. Having said that, there are some patients who have MBL deficiency and have recurrent infections. And there's actually a lot of interest in looking at how MBL deficiency can contribute or modify other diseases. So an example is individuals who have cystic fibrosis, which is an immune deficiency in and of itself, can having an MBL deficiency on top of cystic fibrosis, sort of progress worsening in lung disease. Another modifier is, for example, there are some types of cancer that get treated with chemotherapy. It's thought that having an MBL deficiency might make some patients more prone to have side effects from the chemotherapy that they're receiving for their cancer. So again, some interest in not only MBL as a standalone immune deficiency, but also how it could potentially be modifying other diseases. Okay, now we're going to kind of move on to deficiencies and regulators of complement. And these are a little bit different. So what happens when you have deficiencies in complement regulation, because you're not dampening down that complement activation, your complement proteins get consumed and their quantity is low. And when your quantity is low, it in some way mimics as if that complement protein were deficient. So for example, we talked before about complement regulation, how there's this regulator called factor I that works in concert, sort of partners with these cofactors, factor H, a factor called membrane cofactor protein. When these partner together, they can break down C3b into these smaller fragments that make them inactive. So if you have a deficiency in one of these complement regulators or cofactors, it allows C3b to go around and continue to kind of go back and amplify and activate complement. And so C3 gets consumed. So it's not surprising that C3 levels are going to be low and that these patients are going to present very similarly to patients who have other complement deficiencies like Neisseria infection. And then these patients are uniquely susceptible to a type of kidney disease called atypical hemolytic uremic syndrome. And then there have been some genetic alterations in these regulators that have also been associated with a condition called age-related macular degeneration. How those all really link up isn't entirely clear, but definitely there's some thought by not having proper complement regulation that somehow having that ongoing complement activation, again, is harming some of our own tissues like the macula in the back of our eyes, as well as the kidneys in the case of hemolytic uremic syndrome. And then the last sort of deficiencies in complement or regulators of complement that we'll go over is C1 inhibitor. C1 inhibitor could really be its own IDF lunch and learn. As I mentioned before, C1 inhibitor is probably one of the more relevant, clinically relevant deficiencies in complement. And the reason for that is that there are many people who are affected that have a condition called hereditary angioedema. We talked before about how that C1 inhibitor helps in kind of breaking down that C1 complex for the classical pathway, as well as that mannose binding lectin complex for the lectin pathway. And so what happens is it's not actually related to those complement activation pathways, but by virtue of having these complement proteins, due to its activity on a totally separate pathway, it results in the formation of mediators that cause swelling of tissues like hands, skin, and the mouth, like in this picture that you see here. And there are two types of hereditary angioedema. There's type 1, where you have low levels or low quantity of C1 inhibitor, and there's a type 2 where the number or the amount of C1 inhibitor is normal, but it doesn't function properly. And then the other kind of unique deficiencies in complement regulators, if you have a deficiency in complement receptors 3 or 4, it can actually cause an immune deficiency called leukocyte adhesion deficiency. These patients often have severe infections. We talked about how complement helps recruit neutrophils and monocytes to sites of inflammation or infection. Well, that doesn't happen. And so sometimes these patients will have what we call kind of cold abscesses. Another hallmark feature of leukocyte adhesion deficiency is a delayed separation of the umbilical cord in children who are born with leukocyte adhesion deficiency. And the way to look at that or diagnose it is actually through a specialized test called a flow cytometry test. And then the last deficiency that we'll go over is the CD59 deficiency, which causes a condition called paroxysmal nocturnal hemoglobinuria. Again, only can be diagnosed by a specialized test called flow cytometry. Okay, now I'm going to end with just, again, some very generalities around the management of complement deficiencies. So in addition to finding perhaps a provider that you can partner with and entrust, to manage your complement deficiency, you want to have conversations with that provider about, what are the concerning signs I need to monitor for? What degree of vigilance do I need to have for infections so that I can have not only aggressively recognize it, but get on the appropriate treatment for that infection, whether it be like a sinus infection, a pneumonia, or the Neisseria infections like we talked about for some complement deficiencies. And then vaccination. So for some of these encapsulated bacteria, and for Neisseria meningococcal disease, there are actually vaccinations that are available. And by boosting our immune response or antibody responses through the vaccines, it actually offers us some protection against those infections. So H influenza or Hib, the pneumonia vaccine that's actually available for both children, and there are boosters that are available for adults. And the Neisseria meningococcal vaccine, I would say by far the most important vaccinations to get for patients who have complement deficiencies. The other is antibiotic prophylaxis. Now, it's not universally applicable to all patients with complement deficiencies, but again, worth a discussion with your provider. Depending on the type of infections you may get, antibiotic prophylaxis can sometimes reduce the frequency or severity of the infections you may have. And then there's disease-specific medication and management, and this could be one of several things. So for example, when we talked about how some complement deficiencies increase the risk for autoimmune conditions like lupus, well, I'm still going to treat that patient with lupus similar to someone who has lupus and does not have a complement deficiency. But we talked a little bit about how some of those complement deficiencies and deficiencies in their regulators really produce unique manifestations. So for example, like that paroxysmal nocturnal hemoglobinuria, well, there are actually unique treatments that are available for those diseases that patients may be on if they have that particular complement deficiency. So those are things you want to explore with your provider as well. And there's replacement therapy, and replacement therapy can mean one of several things. First, for some, I would actually say for C1 inhibitor, it's probably the most common. You can actually replace C1 inhibitor and prevent those attacks of the angioedema, the pictures I showed. There are some complement deficiency patients who are on IVIG replacement. So they get on IgG infusions or subcutaneous injections. It's thought by, again, we talked about how complement can boost our humoral immune responses. So maybe by way of giving more of our humoral immune response, you can kind of work in concert with whatever complement is there to help in our defense against infections. Again, not a universal application for all complement deficiencies, really based on an individual case. Because a lot of our complement proteins live in our plasma or blood, there are some, and I would say very rare instances where giving like plasma infusions for patients who are deficient in certain complement proteins may be beneficial. But again, those are very unique situations for which those are applied. All right, so some take-home points for this. First, complement deficiencies are not that uncommon, depending on which complement deficiency that you're talking about. Recurrent sinus and pulmonary infections, those Neisseria infections, lupus, angioedema, and kidney disease are by far going to be the most common clinical presentations that we see with individuals affected by complement deficiencies. And then the diagnosis or the evaluation, not only by getting a really good history, doing a really good exam, but also take advantage of these tests that are available to us to either measure complement levels, so quantity, or measure function. And then management is tailored to that particular deficiency as well as the individual and how they're affected by that complement deficiency. Okay, and with that, I want to thank everybody again for this opportunity. I love talking about complement. And then I think Megan will take some questions and some closing statements, right? Let me stop sharing first.
We sure will. Thank you so much, Dr. Wu. That presentation was very helpful. And even just the way you formatted the information in the pictures, I found really beneficial, especially ones that demonstrate the carrier. Those are always really beneficial and helping explain that to our audience. But I'll go ahead and reshare my screen now.
Mm-hmm.
And as you mentioned, we do have time for just a couple of our audience questions. And then for those who have the opportunity to sit tight with us, I'll also end us this afternoon with information regarding some upcoming IDF events and programs. But the first question I have for you, Dr. Wu, since C2 is not involved in the alternative pathway, would C2 deficient patients potentially still have ability to compensate through that pathway?
Yeah, that's a really excellent question. And so hopefully by talking about the three different activation pathways, everybody was able to appreciate how there are some redundancies, right? And some commonalities in the pathway. And so if you are deficient in an earlier component, there is a possibility of compensation through activation of other pathways, which is why I think sometimes depending on what is deficient, how you may not be prone to certain types of infection. So I think the short answer to that question is yes, absolutely, that could be possible.
And as I'm scrolling, I just want to share a couple comments with you too, Dr. Wu. This was the most
Mhm.
comprehensive and educational in-service on complement deficiencies. Thank you so much. This presentation is what I assume is semesters of learning and years of practice into an hour. This is probably the best explanation of this topic to a layman I have seen yet. So the reviews are in. We're all really excited to have had the opportunity to learn with you today.
I
Um,
appreciate that.
Another question, is lupus more common to develop later in life due to more cell debris slash immune complex buildup over time?
Mm. That's an excellent question. So generally speaking, not in the context of complement deficiencies, lupus is prone to develop later in life. We're not entirely sure. We definitely think there may be a hormonal component. So lupus is something that we consider kind of multiple factors contributing to its risk. One is genetic, because we think there's something that we inherit in our DNA that might place us at higher risk. Also, there are certain populations that are more severely affected, so minority races and ethnicities. And then again, we talked about females of childbearing age. And so it affects women more often than men, particularly women who've undergone puberty. So maybe some hormonal component. And possibly, I don't think if it's necessarily been proven, but maybe some kind of buildup and introduction of cell death and debris over time. I've always thought about it as a hormonal component, but could potentially be a contributor as well.
Thank you. This person you may not be able to speak to specifically, but I'll ask
Mhm.
in case you have some global feedback for them. But they've shared that they've got ongoing CVID, low IgG, low absolute lymphocytes. Just as an example, complement C4 was 16 to 20, but they have seen a drop to 0 for the last year. They're wondering if there's any specific reason it could suddenly drop. They mentioned having nonspecific connective disorder, but not lupus.
Yeah, that's a tough one. So let me reiterate that. So common variable immunodeficiency, low immunoglobulin, low lymphocytes, and a complement level that was complement protein C4 level that was kind of hovering around low normal, and then all of a sudden went to 0.
Hmm.
Um, Reasons for that could be multiple because we talked about kind of how either through the mannose binding lectin pathway or the classical pathway, which both have complement protein C4 involved, if there's something that's activating it upstream that can cause your C4 levels to drop. Now, to drop to 0 is a little bit unusual. So there's got to be something that's activating the complement, whether it be like immune complexes that are circulating around, some sort of microbe, although I would think if there was an infection, it would be a little bit more overt. So I would definitely look for evidence of complement activation. The other thing is if it's only been done once, I would definitely repeat it. So one of the things about complement is it's very finicky. It likes to be high maintenance. And so it's very temperature sensitive. So for example, if a blood sample where you're looking at complement stays out for hours, even just at room temperature, it causes this just kind of uncontrolled complement activation for everything to drop. And so if I were seeing a patient like that, I would think about repeating the C4 level, looking for reasons why there might be complement activation. Think about doing some of those pathway functional testing that we went over to help us better hone in on perhaps what's getting activated.
And we have time for just one more question. We had an audience member ask, are there any research studies for genetic hypocomplementia patients?
Yes, so I think for resources and opportunities, and I'm sorry, I didn't put a slide in there, but of course, the IDF is always a great resource. For studies that are available, research studies, one of the best ways to look at that is clinicaltrials.gov. Whether it be studies related to the evaluation of complement deficiencies, how it might cause certain conditions, or even treatment, clinicaltrials.gov will be a great place and a reliable place to go.
Thank you so much for that. In order to be timely today, we'll go ahead and say another thank you to Dr. Wu for her time. For those who can hang on, I do have a couple of more slides just with some general updates about events and programming. But thank you again, Dr. Wu. If you need to leave, you can go ahead and do so. All right. So thank you for those who submitted questions. Be sure to check out our website at primaryimmune.org, where you'll be able to find more resources, events, support services. Everything on there is free to access in print. And we could also help you find an immunologist or PI specialist in your area using the Clinician Finder. And for those of you who we did not have a chance time-wise today to get to your question, we also have the Ask IDF resource, which is another avenue in which you can submit that question. As a note, all of our Lunch and Learn and webinar recordings can additionally be found on our YouTube channel. I'm also happy to share today that applications are currently open for the Immune Deficiency Foundation's Research Grant Program. So we support well-defined research projects with a specified benefit for improving the treatment, health, disease management, or diagnosis of people with PI. Researchers may apply for up to $50,000 for 1 year towards a specific project, and those applications are going to be due by February 29th. There is a link at the bottom of the slide that you can visit to learn more and apply. As you guys also fill up your 2024 calendars, I ask that you add these two dates, scribble those in as well. So we have two Ask the Expert style webinars coming up, one next week and the next in February. This format will include brief presentations from some of our top clinical experts, where a majority of our time will actually be spent reviewing questions submitted by our PI community members. So you can come learn more about the management of multiple conditions as well as PI and fatigue. The events platform at the bottom of the slide, primaryimmune.org/events, will be the way that you can see information for both of those offerings. Registration is also open for the 2024 PI conference. This is going to be hosted in person, and it's at the Sheraton Grand Chicago Riverwalk from June 20th to 22nd, with our early bird registration deadline being March 31st. You can visit the webpage at the bottom to register. And for those who are interested, we also have a scholarship application open. So for this must be individuals diagnosed with PI in order to be eligible to apply. And you can visit our conference landing page to apply by January 31st. You can also email events@primaryimmune.org for additional information. And last but not least, information about Advocacy Day. So we'll soon be able to share that date for our 2024 Advocacy Day. And this is an opportunity for members of the PI community to travel to the nation's capital and meet with members of Congress. So super exciting. We have a focus on accessibility with this offering. IDF will actually reimburse the costs associated with attending the event, including travel to and from DC, hotel stays, and meals. And the number of participants is going to be limited and selected through an application process. So stay tuned for more information on that one. I also want to give a special shout out again to our partners who made this offering possible. It's their generous support that allows our educational programming and community services. And a big thank you for your participation. And again, to Dr. Wu. We look forward to seeing you all virtually at one of our events this winter, or potentially in person at our 2024 conference this summer. You guys all take care, and we hope you have a wonderful rest of your day.